Okay, hello everybody. So this is another lesson of your developmental biology module, and I am Dr. Mirko Pegoraro. So in today's lesson, we are going to talk about the development of neuronal cells. So the lesson today is neuronal cell development. Where are we in the module? So in the module, in the last lesson, we talk about the paraxial mesoderm. In the next lesson, we are going to talk about the intermediate lateral plate mesoderm as well as the endoderm. This lesson of today link very well with the anterior posterior axis formation and neurulation lesson, as well as with the paraxial mesoderm lesson. So today we are going to talk about the development of the brain, the central nervous system, so the brain and the spine, as well as the peripheral nervous system. We're going to talk about neurogenesis and brain growth, neural cell, neural crest development, as well as the axon growth and the synapses formation. So the cells that build our nervous system, and actually the nervous system which vertebrate, derive from neuroepithelial cells. These neuroepithelial cells are multipotent, and they are the progenitor of the two groups of cells that build up the nervous system, the glial cells and the neurons. The glial cells are very proliferative, so they can divide very quickly, especially obviously during the development, and they can produce other glial cells as well as neurons. They're also important because they provide structure for the neurons to grow and move. The other group of cells that we are going to talk about today are the neurons. The neurons are the ones that transmit electrical potential, so they transmit information biochemically from one place to another in the body. Here is a picture of a neurons. In the neurons, we can recognize a body cell. So the body cell is called the soma, and is where we find the nucleus. The body cells, the body of the neuron, also have dendrites. Dendrites are an arbor of outgrowth which connect other axons. The axons are very long outgrowth or protrusion from the body cells that transmit the information. So each neuron receiving from information via the dendrites, so biochemical information via the dendrite, and transmitted via the axons. And of course, where the axon meet dendrites, so the axon from one neuron meet the dendrites from another neuron, we have a special connection between the two that is called the synapses. We also have synapses between axons of neurons and other target tissue. Or other target tissue could be muscle cells, so neurons in the muscles around the body, or glands around the body, so they control the production of different hormones. We can also have neurons that transmit information back to the body. So imagine information traveling from the senses, from the eyes, for instance, back information that travels from the eye through the neurons back to the central nervous system in the brain. Now, one particular and very important characteristic of axons is that axons do have a myelin sheath. The myelin sheath is basically made up of special cells that are the oligodendrocyte in the main nervous system, so the central nervous system, or glial cells like the swarm cells in the peripheral nervous system that form different layers, so they wrap around the axon and they form different layers to insulate the axons, so that the biochemical electrical information passing through the axon is not dispersed before reaching the end of the axons. In the same way, we isolate the electrical cables in our house so that the electricity doesn't disperse in our walls, or we don't get shocked when we touch them. So the axons need to be insulated so the information is not lost along the way. The brain is built with different layers of neurons. But these different layers of neurons are always organized in more or less the same way. So here in this cartoon, you have how a typical layer of neurons is organized in the brain. So on the left-hand side, there is the lumen. And if you remember previous lesson, we say that when the neural tube is formed, we have in the middle obviously is empty, and that empty part of the tube is called the lumen. So near the lumen, we have what is called the ventricular zone. The ventricular zone is also called as proliferating zone. Here we find most of the glial cells that are proliferating and dividing. More externally, we find the intermediate zone, also called the mantle. And in the intermediate zone, we find glial cells that have been migrating from the ventricular zone, as well as neurons that are migrating from the ventricular zone or actually being produced directly in the mantle zone. More externally, we find the marginal zone, and in the marginal zone, we find mostly the projection from the neurons, so the axons. So it is very interesting because the marginal zone, which is rich in axon, is also referred to as the materia, the white matter. It is called white matter when people look at the brain because of this not being rich in soma of neurons. This in contrast to the mantle zone, which is rich in soma of neurons, and it's always often referred to as the gray matter. If you look on the maturing nervous system along the spine of a vertebrate, you find that at one point the mantle assumes a characteristic shape. It is called a butterfly shape. You can see here in this cartoon. And this butterfly shape of the mantle is surrounded by the marginal zones. Near the center, we have the ventricular layer, and in the middle, we have the lumen. Now, it is interesting to notice how the dorsal portion of the mantle actually receive information from sensory neurons. So sensory neurons that collect information from the hands, for example, for the touch, or for the taste, or the sound from your ears, or the vision from the eyes, are collected on the dorsal part. All this information are collected on the dorsal part of the spine, of the mantle. While the ventral part of the mantle, in the ventral part of the mantle, we find motor neurons. So neurons that originate from that region and innervate a different part of the body, permitting contraction of muscles. This organization of layers is maintained throughout the nervous system. This simple organization, if you want, basic organization of neurons, is found throughout the spinal cord. And you can see here we have the ventricular zone with most glial cells reproducing, the mantle or intermediate area where we have the body of neurons, which is also called the gray matters, and more externally we have the projection of the neurons in the marginal zone, which is called the white matter. However, there are other areas of the developing brain that maintain the same organization but with some modification. I want to discuss with you two in particular. One is the cerebellum, and the cerebellum is the part of the brain, the posterior part of the brain that attach from the base of the brain that attach to the spine. And what we recognize here is that the same kind of organization, however, there is a layer of cell that have been produced initially from the ventricular zone, but then migrate very externally and produce a layer of external granular layer, is called, of this granular cell. And this granular cell start to reproduce, start to divide because of BNP signaling. After that, they start to divide. This cell migrate internally and form an internal granular layer. You can see them here. At the same time, the ventricular zone start to produce multiple different kinds of neurons, and in particular one group of neurons which are called the Purkinje cells. The Purkinje cells are found externally, near actually the external granular layer, and these Purkinje cells produce SHH. SHH sustain cell division of the external granular layer. One other important factor or element of these Purkinje neurons is that they build gigantic dendritic arbor on top of them. So they have an enormous dendritic structure on top of them, forming up to 100,000 synapses, so 100,000 connections per each neuron, per each Purkinje neurons. This cerebellum is associated mostly with the control of balance and control of movement. So all this information that we require to control, for example, our own balance and movement, but imagine other animals too, including birds and reptiles and other mammals like lions and so on. All this information pass through these Purkinje cells. Another particular interesting part of the brain in terms of layering is the cerebral cortex. The cerebral cortex corresponds to the external part of the brain. This is typical of mammals. And this part of the brain is what you think of when you actually think of a brain. So if you see pictures of brain or cartoon of brain or movie where they show the brain of a person, that's the part of the brain that you always see or imagine, which is the cerebral cortex. Now, the cerebral cortex is particularly important because some of the neurons from the mantle actually migrate more externally, forming a layer of neurons called the cortical plate. This cortical plate is also referred to as neurocortex or neocortex. The neurons in the neocortex express a transcription factor which is LHX2, and this is associated with producing more genes, controlling the expression of more genes related to function, related to what we would call higher function like thinking, but also associated with receiving information and processing information from our senses. What is particularly interesting to notice is that there is not one single layer in the neocortex. In fact, the neocortex is built up to 6 layers in mammals. So it becomes very, very complex, and is one of the reasons why, for example, our brain has the shape that it has. So the neocortex will stratify up to 6 layers of neurons, neuron cell body, and all these different layers have different function, they have different properties, they have different kind of neurons, and they form different connections. So it is a very extremely complex structures which control a very important part of human and animal behavior. So it is worthy to spend an extra slide try to understand how the neocortex is actually built up. And it is built up from the foundation up, if you want. This picture look like complicated, but stay with me, and we go through each element of the picture. So you can see here we have time. Time representing neurogenesis, and in this case we are talking about neuron formation in the neocortex. So initially what we have are neuroepithelial cells. So neuroepithelial cells, they proliferate very quickly, they proliferate a lot, and they produce more neuroepithelial cells. They produce these neuroepithelial cells in what is called the ventricular zone. The ventricular zone is the area that is more adjacent to the lumen. At one point, this ventricular neuroepithelial cell start to produce one kind of glial cells. And in fact, here we will find three different types of glial cells. The first type is the ventricular radial glial cells. The ventricular radial glial cells maintain connection, you see this protrusion, with the basal part, or if you want, the lumen part of the developing nervous system. But it also has protrusion through the external part. So if you want, these ventricular radial glial cells remain connected between the internal, and make a connection, a bridge if you want, between the internal and external part of the developing brain. Some other glial cells are formed on later on a more external layer, which is called the subventricular zone. The subventricular zone and the ventricular zone together form the germinal strata. It is called the germinal strata because this is the layer of the brain that during development produce a lot of cells. Most of these cells are glial cells, but also obviously neurons. So these are the region where the neurons are produced. So the other two kinds of glial cells are the intermediate progenitor cells that you can see here. There are these cells here that are produced directly from the ventricular radial glial cells, and these intermediate progenitor cells do not have much ability to divide. In fact, often they divide only once, and they produce the only product that they can make, the only kind of cells that they can make is neurons. So the IP cells produce neurons, but they don't divide too much, so they divide probably only once, producing these two neurons. The third kind of glial cells are the outer radial glial cells, or ORG. The ORG are also produced from the VRG, but the ORG can divide and produce other glial cells, like more ORG or IP. One characteristic of the ORG, as the name suggests, is that they are more external compared to the ventricular radial cells. So we found the body of the ORG not in the ventricular zone, but in the subventricular zone. Please notice how both the ventricular radial cell and the outer radial cell, and time to times even the intermediate progenitor cells, have this structure that bind themselves, this protrusion that bind to the external part of the developing and growing brain. This structure are important because the neurons that are produced in the ventricular zone or the external zone can use to migrate from the internal part, from the lumen, to the external part of the developing brain. Where this neuron come from? Well, we saw that the neurons come from cell division of IP, the intermediate glial cell, intermediate progenitor glial cells, but also via division of the VRG. The VRG can produce more VRG in neurons or directly more neurons. And the neurons migrate over this protrusion from the VRG, but also from the ORG, from the internal part of the developing brain to the external part to the developing brain. In this case, we are talking about the cortex, and the earlier neurons, so the neurons that are produced earlier, travel the shorter amount, and they produce the deeper layer of the brain, of the cortex, the L6. Then more neurons that are produced later during development travel the most along this long protrusion, and they reach more external layer. So the younger, in a sense, the neuron is, the more they travel away from the internal part of the nervous system, and therefore the more external layer of the cortex. But how is that these neurons travel from this internal part of the developing brain to the external part of the developing brain? Well, one of the main and important signals that permit this movement is reelin. Reelin is produced by specialized cells that are called the causal Retzius cells that are situated externally or in the external part of the developing brain of the layers. Because they are produced there, then they diffuse and form an external to internal gradient of expression and concentration. This reelin bind a receptor on the neurons. And when they bind the receptor on this neuron, they induce through a series of cytoplasmic pathway, they induce expression of disabled 1 or DAB1. Disabled 1 or DAB1 induce the expression of NCAD and F-actin in the migrating neurons. So in this neuron, when they start to express NCAD and F-actin, what we will see is the development of protrusion, or if you want, philopodia, or they allow them to extend the body of the neurons toward the direction of travel. Moreover, because they are expressing NCAD, they are able to attach to the surface and actually pull the rest of the body in. So imagine that these are like hands. So because of that one that allow the expression of F-actin, the hands that first is closed and present the neuron that is now moving is able to produce this finger out, which are full of actin. Then the hand is expressing encoding, and the encoding allow the hand to bind, which is the neurons, allow the hand to bind to the substrate. And because of the F-coding, the neurons now is able to close up, and in doing this, the body of the neurons, here, number 1, is pushed up toward the direction of travel. Now, toward the end, when the neuron reach its final destination, that one induced the expression of integrins. You can see them here in green. And integrins allow the neurons to bind to the matrix that is near the causal cells, which is rich in fibronectin. At this point, the neurons experience very high concentration of reelin. This high concentration of reelin had the opposite effect on disabled 1. If relatively lower concentration of reelin was activating DAB1, very high concentration of reelin now suppress it. So when we have suppression of DAB1, we also lose F-actin, so we lose the finger of the neuron, and we also lose NCAD. So the neuron doesn't bind anymore to the surface, and it can stay put where it is, forming, being incorporated to the new layer, integrating itself with the new layer of neurons. And so we can form the first layer, and then the second layer, and then the third layer on top of that, so goes on.